Hi there, Year 11. In this video, we're going to look at sampling techniques and assess the strengths and weaknesses of various techniques. But before we jump into that, I wanted to talk quickly about the koala. Now, the koala is obviously a native Australian animal that you should be quite familiar with, and koalas are becoming endangered across Australia for a whole variety of reasons, and in some areas they're on the verge of becoming extinct. In fact, estimations are that in a lot of areas of Australia, koalas are actually functionally extinct already. Now, functionally extinct means that there's not enough koalas left in the population for that population then to regenerate and become back to the level of biodiversity and the level of individuals needed to sustain that population long term. So it really is a precarious position that the koala is in. Now, the reason for this is there's a multitude of reasons. So koala populations have basically been in decline since white men arrived in Australia. And the koalas were hunted for their fur in the late 1800s to the early 1900s. So, not that long ago, only just over 100 years ago, did we stop sort of hunting koalas for their fur. Now, one of the main reasons koalas are in danger is because of habitat destruction. Over 80% of their original habitat has been destroyed to make way for housing and industrialization. And koalas are unique in that they're perfectly suited for the individual trees or the individual forests that they live in. They're not that great on the ground. They're very slow, which makes them very susceptible to domestic dog attacks and other predators getting them. But if they're up in the tree, then they can usually survive there and thrive there for quite some time, provided the trees are there for them to live in, which is, again, part of the problem with the koala population. Now, the recent bushfires at the end of 2019 had a really devastating effect on a lot of the koala populations around New South Wales in particular. Again, because koalas, they're slow, they're lumbering, they live in the trees. If the trees are on fire, they have a really hard time getting away from that fire. And the other reason for koala population decrease is to do with road accidents as well. Now, it's estimated that there's as few as 43,000 koalas left in the wild, which is really quite a drastic reduction in the total number of koalas that were there up to 100 years ago. Now, why is this important and how does this relate to sampling technique? Well, the reason why we know these numbers, or we know these figures, is because of the sampling techniques that are used to estimate the population of koalas in various areas. So we have a pretty good idea of the number of koalas in certain forests or certain parts of Australia because we do certain sampling techniques to count them and to have an educated guess on how many there are. And that leads us into this idea of sampling, and sampling really is about measuring populations. Now, when it comes to sampling, there's four main types of sampling that we're going to talk about. There's point sampling, transects, quadrats, and then capture, mark, recapture. Now, transects and quadrats you should be familiar with from our excursion to the farm to Windermere. But point sampling, capture, mark, recapture, they might be new things for you to understand, and certainly we'll cover the transect and quadrat as well in this video. So this is a map here looking at point sampling, and what's happened here is we've got a map of an area, and each of these red dots is a point that the ecologists are going to go to and take a sample of the organisms living in that particular ecosystem. So these points might be chosen for a specific reason, or they might be randomized as well. But the idea is we're going to have different samples taken at these different areas here, and by having this a bounce of points that we're going to sample from, we should get a good estimation of the total amount of wildlife in that particular area. Now, when it comes to point sampling, we go to those points and we count the organisms at those points. So it can be used to determine the range of organisms in an area. So if we're looking at, say, an example like the Windermere farm, if we have 20 different points we're going to go to and take samples from, we can get an idea of the range of organisms found across the whole farm. And that can also help us make some calculations to determine how common those organisms are in that ecosystem. Now, the good thing about point sampling is it's really time efficient. It's an efficient way to do this sort of sampling and get a good estimation of the wildlife in that area. And it's not disruptive to the ecosystem either. We're not taking things out. We're literally just going to the point on the map and counting what we see. Now, one of the downsides of that is that rare organisms might be missed if there's a particular let's say an animal that's not as common in that area, or a plant that's only found in a certain part of that farm, for example, and we're just going to certain points, there's a strong chance we might miss that as well. And it's really only effective for slow or non-moving organisms. So an example could be with the koala, which is what they do a lot of with koalas, is this point sampling. They'll pick points in a bush, they'll go out and count the koalas in the tree and use that to get an estimate of how many koalas there are in that area overall. All right, this here is a picture of a transect, and a transect is what we tried to do out at Windermere, where we we pull out the tape measure, the straight line, and then we count and draw the different trees, in our case, that are on that particular transect. Now, this is an example of a high-level transect, and the main thing to keep in mind here is it's going to show this distribution of the flora, of the plant life, along that transect. So basically we start here and we walk for 100 meters in a straight line, and then we draw whatever vegetation we see there. Now you'll notice, just looking at this transect, that if this is a particular forest that we're looking at, what it will tell us is that when we start the transect we have fairly low-lying shrubs and bushes. But when we get about 50 metres to 60, 70 metres into that forest area, we start to get some of these bigger trees starting to develop and starting to show up as well. So it gives us a good idea of the distribution of that plant life in that particular area. Now, this transect will also measure the height of the trees, and there's a way we can do that using trigonometry. And in this case, they've even included these dead trees here as well as part of the wildlife. They're not always included in transects, but in this case, they have included them. And again, they can give us some really valuable information on what's going on in that ecosystem. So transects are basically counting organisms along a straight line and sampling those organisms as well. So it's really useful for determining changes in a community or changes in a landscape, and they can be helpful to determine how common an organism is, particularly with plant life as well. Now, again, it's time efficient. There's minimal disturbance to the environment, which is always a great thing. But just like the point sampling, there can be the opportunity for rare organisms to be missed. And it's really, again, only suitable for slow-moving or even non-moving organisms like trees. This here is a picture of some people doing some quadrat sampling. And again, this is something we did at the farm, where we randomly threw out an indicator or a marker, and we built our quadrat around that and counted the proportion of vegetation in that area. Now, it can be used to count individual flowers, like these individual flowers here in this case, or it might be used to give an estimate of the amount of grass cover. So rather than counting the individual blades, we might say there's 80% grass cover in that quadrat. And if we do enough of these quadrats, we can get a good sample of what that whole area is likely to be. Now, quadrats are using squares on a map, and we use those to count the number of organisms that fall within that square wherever it ends up lying. Now, it's useful in identifying a range of organisms in an area and also getting an idea of how common they are, but it can be very time consuming to do, particularly if you want to do it well as well. And again, it's really only good for slow moving or stationary organisms. So probably not going to be great for counting kangaroos, but would be really great for counting kangaroo bushes, for example. And the last technique we're going to talk about, sampling technique, is called mark capture or capture mark recapture. And what we have here is a picture of some currawongs in the tree, and if you look closely you can notice these bands around the legs of the currawong. And this is an indication that these currawongs at some point have been captured. They've been marked or banded in this case, and then they've been released and recaptured perhaps later on to get some data on the population of the currawong and how many there are. So it's a really effective technique for counting and sampling the total number of animals in an ecosystem, and is somewhat invasive to the animals, but generally the currawong, once they're released, they don't realise they've got these bands on, and they can live their normal happy life with those on, and we can collect some information on the currawongs by looking at those bands. So animals are captured, they're marked and then they're released. And then after a set period of time, the population is then resampled. And we use the same capture method. They might use nets, they might use other ways to capture, in this case, the birds or the currawong. And based on how many they recapture, how many of those they've marked that they then recapture, that can give us an idea of the total number of organisms or birds or animals in that particular area. So it's useful in determining the total population for mobile species, so birds, possums, kangaroos, these sorts of things. You might have even seen videos of sharks being tagged or whales being tagged with similar sort of sampling methods. Now, it's very time consuming to do well. It takes a lot of effort to catch the organisms, and it's not really suitable for the slow moving organisms because we can use much less invasive systems and sampling systems to sample slow moving organisms. And you need to be careful that whatever method you use doesn't interfere with the behavior of the animal or the movement of the animal. The last thing we want to do is put something on the animal that then causes that animal to have to become more likely to be attacked by another animal or to be not unable to support itself. Now, what I'd like you to do now is I've linked a video just underneath this one in the OneNote that's going to show a technique of capture-recapture for ants. It's a really interesting way they do it with ants, and it talks a little bit about some of the maths behind what they're trying to achieve. And then on the activities part of OneNote, there is also a question sheet with some information on all of these sampling techniques as well.